Gluconeogenesis is the process by which certain types of cells of our body have the ability to actually synthesize glucose molecules by using non-carbohydrate precursor molecules. So these precursor molecules include pyruvate molecules, lactate molecules, glycerol molecules, and amino acids. So these are the major types of non-sugar precursors that we use inside our body to synthesize glucose via gluconeogenesis. Now, only specific types of cells in our body are actually able to undergo gluconeogenesis. So these include liver cells and, to a small extent, kidney cells, because it's the liver and the kidneys that are responsible for actually regulating and maintaining the proper glucose level in our blood. So liver cells, hepatocytes, and kidney cells are able to actually undergo the process of gluconeogenesis, and cells like cardiac muscle cells, skeletal muscle cells, and brain cells cannot actually undergo gluconeogenesis. These cells depend on liver cells and kidney cells' ability to actually create these glucose molecules, dump these glucose molecules into the blood plasma, and then the blood plasma brings these glucose molecules to the cells like the cardiac cells, muscle cells, and brain cells of our body. Now, let's briefly discuss where we actually obtain these non-sugar precursor molecules. So we know where pyruvate comes from. That comes from the breakdown of glucose. So when we don't have enough glucose in our body, we can actually take that pyruvate and essentially reverse the steps of glycolysis. Now, that's not exactly true, and we'll talk more about that in just a moment. But basically, gluconeogenesis is actually the process by which we take the pyruvate and turn it back into glucose. Now, what about lactate? Well, we know if we're exercising vigorously, our skeletal muscles will begin to produce ATP to actually contract our skeletal muscle. And when that happens, eventually we're going to run out of oxygen. So when the rate of glycolysis is much higher than the rate of oxidative phosphorylation that takes place in the mitochondria of our cells, in that case, we're going to begin the process of lactic acid fermentation. And so these skeletal muscle cells produce lactic acid, which dissociates into H+ ions and lactate, and that's where we obtain the lactate from. Now, eventually, that lactate enters our blood plasma and makes its way into our liver cells, and the liver cells transform that lactate into pyruvate by the action of an enzyme we call lactate dehydrogenase. And once that transformation takes place, only then can pyruvate actually begin the process of gluconeogenesis. So lactate itself doesn't actually enter gluconeogenesis. It must first be transformed into pyruvate before it can actually begin the process of gluconeogenesis. Now, what about glycerol molecules? Where do we get glycerol molecules? Well, glycerol molecules are components of fats. More specifically, they're components of triglycerides. So that's the major type of fat molecule that exists in adipose tissue. And so when we actually need glucose in our body, when we can't get the glucose from food, our fat cells, the adipose tissue cells, basically begin breaking down these triglycerides to form fatty acids and glycerol molecules. Now, fatty acids cannot actually be used to form glucose molecules, and we'll discuss why in a future lecture. But the glycerol molecules can be used to form the glucose, and so the glycerol basically makes its way into the blood plasma and travels to our liver cells, and once inside these hepatocytes, they undergo a two-step process. The first step is catalyzed by glycerol kinase, and we use an ATP molecule, and so we transform glycerol into glycerol phosphate. And in the second step, that is catalyzed by glycerol phosphate dehydrogenase, we basically use an NAD+ molecule to oxidize the glycerol phosphate into DHAP, so dihydroxyacetone phosphate. And it's this molecule that enters the process of gluconeogenesis and begins the conversion of this molecule into glucose. So we see that pyruvate enters gluconeogenesis as pyruvate, lactate enters gluconeogenesis as pyruvate, and glycerol enters gluconeogenesis as DHAP, dihydroxyacetone phosphate. Now, what about the final type of precursor molecule, amino acid? So, where do we get amino acids from? Well, typically we get amino acids from food products. So if we ingest protein, we break down the protein, and that's where we get those amino acids. But under starvation conditions, we can actually obtain the amino acids by breaking down our own proteins found in the skeletal muscle tissue. And in that case, we actually deteriorate the skeletal muscle by breaking down the protein, but we can use those amino acids that we obtained by the breakdown to basically form glucose molecules. Now, certain amino acids are transformed into pyruvate, and so they enter gluconeogenesis as pyruvate molecules. Some of them can actually form DHAP molecules, and so they enter gluconeogenesis as DHAP molecules and eventually produce the glucose that our cells actually need for energy, for ATP creation. Now, we still really haven't answered a very important but very simple question. Why does our body actually need gluconeogenesis? Why can't we depend solely on the food for our sugar source? Well, that's because we can actually store a limited amount of sugar inside our body. So to be more specific, in our fluid, so places like our blood plasma and the fluid found in the cells, so cytoplasm, and fluid found in our tissue, in these areas, we can store a maximum of about 20 grams of glucose. Now, in our glycogen stores, we can basically store about 190 grams of usable sugar that can be readily used and can be readily accessible by those cells. And so together, 20 grams in the body fluid and 190 grams in glycogen, we essentially store about 210 grams of sugar. Now, on a daily basis, an individual that, let's say, is not an athlete, needs about 160 grams to actually survive. And 75% of that is actually used by the brain cells. So about 120 grams of the 160 grams is used by the brain. The rest is used by other areas of our body. Now, of course, if you're an athlete, for example, if you're a swimmer and you swim, let's say, a double practice, and that adds up to, let's say, 10K, then obviously you need many more grams of sugar. So those individuals need about 500, 600 grams of sugar. And so what we see is, if gluconeogenesis did not actually take place inside our body, the individual would only be able to actually survive for a little longer than a day. Because once the reserves of these sugars actually runs out, our cells have no way of creating new sugar molecules from these non-sugar components, and in that case, we essentially die. So after a little over a day, we would run out of our glucose supply and our cells would not be able to actually continue functioning. And if we don't ingest the food, we would essentially die. So that's why gluconeogenesis is so crucial, so important, because it gives our body the ability to actually produce these sugar molecules needed by our cells to form ATP from non-sugar molecules such as protein, so amino acids, fats, the glycerol molecules, as well as the lactate and pyruvate molecules. So now let's take a look at gluconeogenesis. So I said earlier that gluconeogenesis is essentially the process by which we transform the pyruvate molecules into glucose. And remember that glycolysis transforms glucose into pyruvate. So at first, it might seem like gluconeogenesis is simply the reverse of glycolysis. Now, that's not actually true. And gluconeogenesis is not simply the reverse of glycolysis. Why? Well, because glycolysis is a very exergonic process. So remember in our discussion of glycolysis, we said that when 1 mole of glucose is broken down inside our body under physiological conditions, that releases about 96.2 kilojoules per mole of energy. And so the breakdown of glycolysis is an extremely exergonic process. It takes place spontaneously and irreversibly. So what that means is, if gluconeogenesis was simply the reverse of this process, then every time 1 mole of pyruvate is transformed into 1 mole of glucose, that would require an input of 96.2 kilojoules of energy. And so what that means is gluconeogenesis would be a very, very inefficient and expensive process. And so gluconeogenesis, for that reason, cannot be simply the reverse of glycolysis. So once again, gluconeogenesis is essentially the conversion of pyruvate into glucose, which is the opposite of what happens in the process of glycolysis. However, gluconeogenesis does not simply follow the reverse steps of glycolysis, and this is because glycolysis is a very exergonic process. It releases an amount that is equal to 96.2 kilojoules per mole of free energy. So we know that in glycolysis, the overall step involves, so we have glucose, 2 ADP, 2 PI, so orthophosphates, 2 NAD+. We have 10 steps that take place that essentially make this process an irreversible process, and we form 2 pyruvate molecules, 2 ATP molecules, 2 NADH molecules, 2 H+, and 2 water molecules. Now, we know that glycolysis takes place in 10 steps. And actually, 7 of these steps are not very exergonic. In fact, in some cases, the individual steps actually require an input of energy. And 7 of these steps are actually at equilibrium, or very close to being at equilibrium. And what that means is, when a reaction is at equilibrium, the Gibbs free energy is equal to 0 or very close to 0. But 3 of these steps, step 1, step 3, and step 10, are very exergonic steps. In fact, these are the 3 steps, so step 1, step 3, and step 10, that essentially make up the majority of the free energy that is released in the process of glycolysis. So remember, in step 1, we take glucose, use up an ATP molecule. In the process of hexokinase, we transform that into glucose 6-phosphate. And this releases 33.5 kilojoules of energy. Step 3, which is the committed step, that's catalyzed by phosphofructokinase. In this step, we transform fructose 6-phosphate into fructose 1,6-bisphosphate. And this is also an exergonic process that releases 22.2 kilojoules per mole of energy. And the final step is also an exergonic process. This is catalyzed by pyruvate kinase. So we have PEP, which stands for phosphoenolpyruvate. In the presence of these two, we basically transform that into pyruvate and ATP. And this releases 16.7 kilojoules of energy. Now, if we sum these values up, so 33.5 plus 22.2 basically gives us 55.7, and then 55.7 plus 16.7 gives us 72.4. So basically, these 3 steps alone release 72.4 kilojoules of energy every time a single glucose molecule is broken down. And that makes up the predominant amount of this total 96.2 kilojoules per mole. So what that basically means is, for gluconeogenesis to actually take place effectively, it must somehow be able to bypass these 3 steps. In fact, that's exactly what happens. As we'll see in a future lecture, gluconeogenesis and glycolysis actually have 7 steps in common. But 3 steps are not in common, and it's these 3 steps. And that's because gluconeogenesis If it simply reversed these steps, so for instance, if we look at step 10, if gluconeogenesis went from pyruvate to PEP by using this specific reaction pathway, it would need to use 16.7 kilojoules per mole of energy. And to prevent this from happening, to make this reaction, this step, an exergonic step, it uses a completely different pathway to transform pyruvate into that phosphoenol pyruvate molecule. So once again, in glycolysis, the 3 irreversible steps release the majority of the free energy in glycolysis. And that means that for gluconeogenesis to be an effective process, to be a spontaneous process, it must actually somehow bypass these 3 extremely exergonic steps. And that's exactly what happens. And essentially, these 3 steps are the steps that are used to bypass these 3 steps here. So let's begin with step number 10. In step number 10, if gluconeogenesis simply followed the reverse of step 10 in glycolysis, we basically transform pyruvate into PEP. In the process, we'd use ATP to form ADP and H. And this would require 16.7 kilojoules per mole of energy. But what happens in gluconeogenesis is a completely different reaction pathway is activated.